Hello there, and welcome back to another Mr. Carter Science special. In this video we'll be looking at the structure of a leaf and the various specialised cells that a leaf contains. As always, keep your eye on the specifications from the syllabus so you can follow along with where you're up to. And we'll start off with a very quick task. I'd recommend that you pause the video, perhaps print off this screen and try this. Can you label all the different tissues in a cross-section of a leaf? Let's look at the answers. So at the top of the leaf, we've got the waxy cuticle. And this is a waterproof layer that prevents two things. One, the entry of water into the leaf when it rains, and two, the leaf losing too much water, which is especially important in very dry conditions. Below the waxy cuticle, you have the upper epidermis, followed by these elongated cells here, which are standing upright. And those are the palisade cells. Now the palisade cells stand upright so that they can gather as much light as possible. You will always lose a little bit of light as it travels through the cell wall. Cell walls absorb a small amount of light. So by positioning the palisade cells in an upright position, we have the minimum number of cell walls between the palisade cells and the light from the outside, which means we get the maximum amount of light being absorbed by the chloroplast in the palisade cells, which can then result in a higher rate of photosynthesis. Below those, we have the spongy mesophyll cells. Spongy mesophyll cells are also cells highly adapted for photosynthesis. Whereas a palisade mesophyll cell is adapted to try and absorb as much light as possible and have the highest rate of photosynthesis, a spongy mesophyll cell is designed to absorb as much carbon dioxide as possible and therefore have a very high rate of photosynthesis. And they do this by having a spongy structure which incorporates different air spaces between the cells. Now these air spaces contain carbon dioxide, and that carbon dioxide will be in a very high concentration from the air and the air spaces, so it will diffuse into the spongy mesophyll cells rapidly, which allows the spongy mesophyll cells to have a very high rate of photosynthesis. So both these layers, the palisade and the spongy, both form the same kind of mesophyll tissue, which is adapted to have high rates of photosynthesis. At the base of the leaf, in the lower epidermis here, lower epidermis, we have two types of cells here. As well as the epidermis cells, we have guard cells, and these guard cells control the opening and closing of this particular gaps, and these gaps are known as the stoma or the stomata. One stoma and many stomata. Stomata is the plural. And the stomata control the movement, or the stomata and the guard cells control the movement of air into the leaf and things like oxygen out of the leaf and water leaving the leaf. So again, pause the video now. Let's see how much you've learned in the last two slides. See if you can write down and fill in this gap fill exercise to explain how a leaf is adapted to its function. So here's the answers. The cuticle, or the waxy cuticle, is a waterproof layer that stops the leaf losing too much water. The palisade cells have lots and lots of chloroplasts to help them absorb light energy and to make photosynthesis happen. Leaves themselves have a very large surface area so they can absorb lots of light energy, and the cells in the leaf need to absorb carbon dioxide from the air, and air gets into the leaf through holes in the bottom of the surface called stomata. Guard cells can close at night to stop the stomata losing too much water. So let's look further at these stomata. Stomata are very useful because they control the movement of oxygen out of the leaf, water out of the leaf, and carbon dioxide and air into the leaf. So oxygen is a waste gas made in photosynthesis, and oxygen diffuses out of the stomata. Carbon dioxide is a gas needed by plant cells to make glucose in photosynthesis, and carbon dioxide diffuses into the leaf through the stomata. Water is a liquid needed by the plant to make glucose during photosynthesis, and lots of water can be lost through the stomata. So during the daytime, the stomata must be open, and they must be open because during the day the chloroplasts are absorbing light energy, there's lots of photosynthesis going on, and the raw material for photosynthesis is carbon dioxide, so you need to be able to absorb that. But the disadvantage of having your stomata open during the day is that you will lose a large amount of water via transpiration. Water is needed by plant cells for them to stay turgid so that they can hold open the leaf and keep the leaf upright so it won't wilt. Water is also needed for a large range of other chemical reactions, and it's also the solvent that we get a lot of chemicals dissolved in. So losing water is a big problem for plants. But the compromise is that you are absorbing carbon dioxide, so at least you can carry out photosynthesis. However, at night there's no benefit to having the stomata open. You will only have the negatives of losing water. Therefore, at night the stomata will close to prevent the water loss, and the guard cells here, we can see there's the guard cells either side, and they will open and close the stomata. So hopefully you have seen in your classroom some real stomata, or at least the imprint of real stomata, under a microscope. If you've done this, you may have taken some sellotape and placed it on the bottom of a leaf, of which there's already some clear nail varnish, and as you pull off the sellotape, you pull off the clear nail varnish and you get the imprint of the bottom of the leaf. And if you look at it underneath a microscope, you can see a number of different stomata. In fact, if you look carefully, you can see the guard cells which surround the whole of the stomata. So how is it that the stomata can open and close during the day and the night? Well, during the day, salts are actively transported into the guard cells, and this makes a very high concentration in the cell. So water moves into the guard cell by osmosis, from a low concentration to a high concentration. And this makes the cells very fat and rigid, and therefore the stomata opens. In fact, stomata have a very thick, guard cells, have a very thick cell wall on the inside of their cells, which resists pushing forces. So when the water and the salts and the water flow into the guard cells, as they try and thicken, it is easier for them to push in this direction. Therefore, they create this hole, the stomata, between the two guard cells. But at night you get the opposite happening. We need to close the guard cells. So the salts that are in the guard cells are actively transported out, and this makes a very low concentration inside the cell. So water will move out of the guard cells by osmosis, from a low to a high concentration, and this makes the cells become thin, less rigid, and therefore the stomata will close. There's our salts moving out, and there's the water moving out. So I hope you've enjoyed this video on leaf structure and the cells and the tissues of the leaf. Please like and share. Don't forget to subscribe and add any comments below. Thank you very much. This has been a Mr. Carter Science Special.